Hello, my name is Mara Aspinall, and I'm very pleased to be here today to talk to you about COVID testing, the key to the beginning, middle, and end of the pandemic. Professionally, I'm Managing Director of BlueStone Venture Partners and Health Catalysts Group, but I'm very proud to be a professor of practice and the co-founder of the Biomedical Diagnostics program at Arizona State University, an advisor on COVID to the Rockefeller Foundation. Today, we're going to talk about COVID testing, specifically diagnostics' role in the pandemic, and then COVID in context. How does COVID compare to the flu and other epidemic and pandemic? And then we'll go back to the future. Where did COVID start and when will it end? Let's start by looking at diagnostics' role and the evolution of testing. When you see this, I'm going to start with the fact that how did testing begin? It was all about symptomatic testing, lung CT and bronchial lavage. Very, very invasive. It moved from there to active disease diagnosis, where we are today with RT-qPCR and antibodies. We then moved to screening. And as we look at screening, there are a number of key technologies: PCR, antigens, CRISPR, breath tests, which now there are one approved in the US, but also COVID sniffing dogs and scratch and sniff. But what does the future look like? The future is all about surveillance. And the technology that is most common today is wastewater, PCR and NGS, but I think in the future we'll also be looking at air quality monitoring. So if we understand that, where were these tests happening? It started in the hospitals, it moved to central lab, then to point of care, and ultimately, as everyone is very familiar with, particularly in the United States, is home and self-testing. So what are these technologies? As I said, it started with highly invasive technologies in an operating room with the lungs, moved to semi-invasive with those nasopharyngeal swabs back of the throat, really the tickle the brain ones. We moved to minimally invasive, the front of the nose, and ultimately where we are today is what I'll call passive with breath and saliva testing. What about the price? We have to follow the money. It started out with more than 1,000, went to hundreds, now to tens. And then I think ultimately we'll get to less than $2 a test. So that gives you a sense of the evolution of COVID diagnostics. So what's so important? Well, time after time, whether it's the beginning, middle, or towards the end, we hope, of the pandemic, everyone wants tests. There are headlines after headlines after headlines talking about the fact that there are either not enough tests or at times too many tests. So what are these tests for? For those in the industry, you all know this, but what we look for are the basics, either the viral element or the immune response. Do I have active disease or did I mount an immune response? At the beginning of the pandemic, if you had mounted an immune response, we knew you had COVID. Now, with the pervasiveness of vaccines, we don't know whether it's immunity from the vaccine or immunity from infection. But those are the key pieces of the system to understand the tests that we have. So how many tests do we have out there? Well, I'm very proud to be the principal investigator with a team at Arizona State University, particularly Nate Wade. He is the co-PI, and we have put together what we call testingcommons.com. And this, I believe, is the largest database of COVID tests in the world. It's not about how many tests, we'll come to that. It's about what type of tests are out there, not just in the United States, but around the world. And not only tests that are there today, but tests that are being developed. I look at them in three different ways. One is the DX targets. The red ones here are viral tests, and the green are antibodies. We then look at platform, RT-PCR, virtually all of the viral RNA, and then we look at lateral flow and ELISA. And where are they coming from? The United States, other parts of Asia, and some from the EU. But let's delve into more details here so you can understand where that is happening. So when we look at that, as I mentioned, it's in testingcommons.com. So we look at those 2,800 tests. Now let's break them down into component parts. We currently have 6 major categories of these tests, from the ones that have EUAs at the FDA, the ones that are laboratory developed tests. The largest category is CE-IVD. We have RUO, or research use development, and a category that many, many people forget are tests that have actually had their EUA revoked. So let's keep going and, as they say, peel the onion here about these tests. I'm going to focus initially on the US FDA EUAs. As you can see here, the vast majority of these tests are molecular. Of that, remember this number, 88% are RT-PCR. We have a few isothermal, a little bit sequencing, and a little bit CRISPR. The next category are the antigen tests in green. And again, remember those. There aren't a lot of them, but they're playing a critical role. We then move on to antibody tests. So many of these were approved early in the pandemic. And then what I call other, which are collection kits, flu or RSV panels, and patient management. Again, very few. So what the discussion is today in early 2022 or spring of 2022 is all about these antigen tests. So let's look at them in more detail. Today, there are 20 EUAs for over-the-counter COVID self-tests. To give you a sense of what it's like in Europe, the over-the-counter tests, there are more than 40 of them. But in the US, we have 20, of which 3 of them are molecular tests. These are all LAMP tests and can be done at home. But the vast majority and the cheapest ones that are most affordable and most available are the antigen tests. All but 2 of them are serial for asymptomatic, so you have to do 2 tests according to directions if you don't have symptoms and 1 if you have symptoms. This gives you a sense of those tests. But what role do they play? And so many people take a look at the CDC website or the New York Times website, and they look at COVID test volume. It's very important that when you look at COVID test volume, that it's not just the gray line here. The gray at the bottom of this graph are the reported PCR tests. These are the ones that the CDC, the New York Times, and other places track. However, what I track, in addition to that, are the antigen tests and those done at home. A little tougher to track for a lot of reasons. Most part that the tests are not reported to any health authorities, and only a small number are even reported to doctors' offices. However, in looking at this, I believe that there are at least 4 times, if not 5 or more times, more home tests done today than lab-based PCR. So when you see those numbers printed, make sure you understand, is it total tests? Probably not. Is it those lab-based PCR tests? And why are these tests so important? It's all about timing. You've probably seen this chart by now, but I'll add a few numbers here, which is typically, if somebody's exposed on day 0, they're not infectious till day 2 or 3, even with Omicron. Symptom onset may not happen till day 4 or 5. Traditional results through a PCR lab test don't come out till day 6 or 7. By that point, 60% of the time that you're infectious is already behind you. So it's key to use these home tests. I know there have been a number of questions about home tests. Are they accurate? Are they accurate enough? This is why it's important, if you have any doubts about those tests, to do those tests in a serial fashion. So that gives you a quick summary of the home test, but what other kinds of testing modalities are there? For schools, the largest type of testing is done by pooled sample. And this gives you a sense of all of the EUAs for pooled sample. There are 2 pooled on the right where you see the number 12. There are 2 tests that have an EUA to put 12 samples together. There are 4 that have 10, 2 that have 8, etc. This is key because it gives everyone the ability to get the test done very quickly and a cost-effective manner with a number of samples. When we were in the midst of an Omicron surge and testing was 30 to 40% positive, not a good place to be. But for now, it's ideal. So where are these tests coming from? Let's look at the companies with the largest number of EUAs. You'll see on the left, Abbott has the most EUAs, followed by Siemens, Roche, and Danaher. But underneath each column, you can see what the makeup of their EUAs are, and how many are in each of the different categories, either molecular, antigen, or antibody, or patient management. So that gives you a sense of it. This was very much focused on the US. I'm going to go now to show you what does it look like in Europe. And you see a different perspective here, where the antibody tests are the most prevalent. Most of these CE-IVD, which is a self-certification process, were done early in the pandemic. About 37% lateral flow, 46% ELISA. Molecular is still here, but about 41% RT-PCR. And you remember for the EUAs in the US, it was about 88% PCR, so very similar. So this is all about tests that can be managed and marketed. What about the tests that have been revoked? And this is critical to understand. You remember the antibody tests are in red? What you see here is almost 80% of all the tests that have been revoked are antibody tests. And this is critical. The FDA has authorized about 250 antibody tests, but 171 of them are now on the do not use. So when you see an antibody test out there, your organization wants to use it, it's critical that you understand not just that it got an EUA, but that that EUA remains in effect. The other caution that the FDA gives us are safety communications. And what you can see here is there are a number of safety communications. I won't go through all of them, but again, very important, and this is often overlooked, to look up the safety communications, the warning letters. Sometimes they're about individual companies, and sometimes, like on the right, safety communications here, warning letters about companies. But sometimes they're about just practices like antibody testing, self-testing, rapid antigen testing, and variants. So this is a key piece of the FDA's approach, and important to keep up with this on a regular basis. So again, we've looked at the core here, but this is mostly US focused today. What about the future? I think it's important to look at the tests in development, not just in the US, but around the world. And remember, we talked about molecular, that PCR was 88 to 90% of all tests. Well, when you look at the future, you see a very different view, where PCR is only 40%, and now you see isothermal, which is mostly LAMP, CRISPR, and sequencing come into about 30% of new technologies. And I'll spend a minute now in talking about those new technologies. You also see fewer new tests coming into the antibody area, where I think we have plenty of antibody tests out there. So just to remind you, what are we looking for in these tests? Viral elements or immune response. You probably are familiar with the fact that we have a lot of technology for the viral RNA and viral antigen tests and immunoassays. The most important change in these tests is that they became automated in central lab and then became smaller by going to a DIY finger stick, less blood, less saliva, less nasal fluids into the small test. That's the state of the art in viral RNA and immunoassays. But what's the state of the art in surveillance? That's all about wastewater. And this looks at just one example of wastewater analysis that was thought of as new. This is actually early in the pandemic, but I use it because it shows how dramatically effective wastewater can be as a surveillance tool. The first 5 bars here, and this is in Tempe, Arizona, were during the lockdown. And what you could see is virtually no change. This is the 0 level of COVID-19 in the wastewater. Just 1 week after lockdown ends, you start to see COVID-19 in the waste. Right after that, more. and by 4 weeks later, you see COVID in the wastewater in 5 out of 6 districts. So this can be a highly, highly effective approach to wastewater, and that's critical to understand. But where do we go from here? What other new technologies are there? So if we look at that, I believe that we will see more and more new technology happen. And it's not just about getting to smaller and smaller. It's also about simplifying the technology, integrating it, and miniaturizing it. The best example here is traditional PCR. One of the biggest challenges is the temperature cycling and the thought that that had to happen in a large instrument. But with unique cooling technologies, we found that you can make those very small into a portable system or even a handheld disposable system. This is a system that has an EUA for a point of care use. We're seeing that in isothermal as well. It's quicker than PCR, but has lower sensitivity. So where else will we go with our technology? What you will see here is on sequencing technology. Sequencing technology itself has changed way before the pandemic to go from huge systems to benchtop systems. But I think we will see new technologies here where we might even have a handheld NGS system through an electrosensing system. It's an hour and a half to result, but think about it. It was 20 years ago that it took a tremendous amount of equipment to do this, and now we might be able to do it in just an hour and a half in a handheld unit. But what about radically different technologies? If we look at these technologies, I think they come in 3 pieces. One is mass spectrometry, which is a protein fingerprint. No sample prep needed. You can use whole blood, and we're looking at positive charged DNA fragments, no reagents, and can be something that virtually every lab knows how to do. We can also look at tetrahertz excitation, a chemical fingerprint, to be used with a throat swab or breathalyzer transferred to a glass slide. The process here in terms of sample prep is more complicated, but it's just 2 minutes to result. And lastly, MRI technology, again, very widely known. Here, again, no sample prep, 2 hours per cartridge. You can probably do about 60 samples a day in 1 unit. These are the technologies that I suspect we will see much more of in the future. So if we take that, where else can we see these radically different technologies? It's in AI. And when we look at AI, I'm going to start with airborne particle detection. This is key. So there are 2 different ways to do it. One is the room pathogens, and the other is breath viral sampling. But the thing that has changed most recently is there is now an EUA for a VOC test, volatile organic compounds, where you breathe into a unit and get your answers very quickly. So challenges, false positives and false negatives, like all sorts of other tests, but still a great breakthrough for this next stage where we may be testing a lot of people, and we want to do that in the shortest possible period of time. What else could we see? Well, maybe it's on your watch that you have a skin detection and a biometric indicators of infection. We're not there yet, but I believe through electrochemical sensing and through imaging, we will make these technologies work. They're working for heart conditions. I believe ultimately we will see them for infectious disease, but not today. So I gave you some examples of where the technology is going, but let's step back a bit and look at the challenges around tests. And I'm going to say it's 3 areas. Number 1, the boom and bust cycle. We don't have time to go through that, but I'm sure you're aware of the fact that we had no tests, everybody ramps up, and now there seem to be extra tests. We need to diversify supply. We now have 17 different antigen home manufacturers, which is great. if this is something that goes on for years, do we need more of those to have healthy competition and lower the price? Everybody looks at it differently, but I think that having a diversification of supply and supply chain is critical. Technology must keep going forward, better, faster, and hopefully more affordable. And we need to ensure that that affordability happens with better distribution, not just for those who can afford to buy the tests. Next is an area that there's been much discussion, which is consistent public health reporting. I talked earlier about the fact that these home tests, as you can see on the second sub-bullet here, reporting is basically non-existent. So I don't know if you've done a home test. Most of you probably have done one or more. What do you do with that result? If you're positive, you might call your doctor, but not in every state, there is not a required system for that doctor to report the positives. What if you're negative? Those are not being reported. Try calling your public health office. They don't have the time or technology or staff to record people individually. And then when we look at public health reporting, do we have one national system? We don't. We have 50 or more systems on public health, so that's another challenge. But lastly is the area that we've all talked about, and I'll share a few slides on this, which is variants. What is the variance? Well, basically, it's the virus fighting back. The virus wants to survive, and by doing it, it knows it needs to change. So we need to track those variants, and we need to understand whether the tests work with them. So let me share a little bit about this. We know the challenge of COVID is about waves of disease. We got down in the US to less than 10,000 cases a day, and today we're celebrating with only 50,000 cases. It's all about these waves. And when we were in June 2021, that's when we were down under 10,000 cases a day, and Delta and Omicron were still to come. So we can't take this for granted. So what is a mutation? You're probably aware of this now, but for the few who aren't, we know that an mRNA strand is almost 30,000 bases long, and that if one or two changes happen to code, it does not change the coding of this protein. But if you start seeing multiple changes, one of three things happen. One is it's so few, it's just a variant of note. It does not change severity, virulence, or mortality. We then move into variant of interest where it starts to change. And then we know what the WHO calls the variants of concern, where there are proven data that shows this has an effect on patients. And it could be infectiousness, virulence, severity, or mortality. It becomes a variant of concern. Well, what does the US look like? Well, if we go back to 2020, you see this original strain in gray on the left-hand side of the chart. There were very few mutations, and we might have gotten a little bit too comfortable with that. We then saw alpha, and alpha became the dominant strain in 12 weeks, and we thought that was fast. Then Delta happened. Delta became the dominant strain in green here, alpha in red here, the dominant strain in 4 weeks. Oh my goodness, and we thought that was fast. Then came Omicron, and it was so contagious, it became more than 50%, the dominant strain in purple here, in just 2 weeks. So when we have a mutation, the question is, what do we need to know? We need to know, does it make tests less accurate, increase cases, increase deaths, make tests ineffective, and does it in fact effect the vaccine likelihood of protecting you? We now know a lot more about Omicron, and reality is it does make some of the tests less accurate, but just a few. And why do I say it doesn't increase deaths? It's because the death rate for Omicron in particular has gone down. The number of deaths have gone up as the number of cases have gone up. However, the percent of deaths have gone down. And why was Omicron so scary? Well, all the rest, alpha and beta and delta, all came from the same original source, but all of a sudden Omicron came very different and looked more like alpha and beta than just a new variant of delta. So it was important to understand where COVID came from, to understand where Omicron came from. I won't go through this in detail, but what you can see is it's critical to understand that it came from, and we'll start with this slide, is the wet market and Hunan. And what we see is there are two markets, the market east and the market west, and virtually all of the cases came from right near the wild animal section in the market west. This was key into understanding that. And what was also key is to know that where it started and where it went to. And what we can trace, not perfectly, but we can't trace the difference between where it went from a bat to some unknown location to then within the market. The challenge here was what looked like a reasonable decision at the time, is that the market was thoroughly disinfected. And it wasn't until 2 months later that we really understood, those in China, that it was important to understand that host in between the original bat reservoir over to the market and to humans. But from there, we can trace exactly what the lineage is and the importance of international spread. So when you talk about international spread, I think it's important to understand, how does COVID compare to other epidemics or pandemics? And it'll take you a little while to look at this chart, but what you can see is here we are today with Omicron, where the death rate is relatively low, but the transmissibility, or the R0, is relatively high. And we've moved from COVID Wuhan to COVID Delta to here we are in COVID Omicron. But where we don't want to go is here, where you have norovirus and measles and mumps, where they're highly, highly contagious, even more contagious than we have today. So what that brings me to is when people say this, they say, yeah, the cold is contagious too, the flu is contagious. So let's compare COVID to the flu. And given where we are now, I compare COVID to the flu across 2 seasons. And briefly, COVID, about 80 million plus. The flu, 2 seasons, 91 million. Hospital admissions, COVID is 3 times bigger than the flu, and the typical time in the hospital is twice as long as the flu. In terms of deaths, this is so sad. I get emotional about this. I have 5 members of my family and friends who have died, almost 1 million people, where the flu is 100,000. And this is 2 flu seasons. And the death rate across the entire pandemic, and I know people say Omicron is just like the flu, it is still 4 times higher than the flu, but if we look across the whole pandemic, what we see is COVID is 10 times higher. And why is this important? Well, it's important because as challenging as that is broadly, it's been disproportionately challenging to groups of color and many minority communities, including Black, Hispanic, and Asian communities. And the US has a higher death rate than a number of years lost than any other country in the world. But what we know is COVID is not one disease. So as I summarize here, I want to emphasize that there are still, COVID we talk about as one disease, but we know that it does not hit everybody in the same way. We know that at least 20% of people, I believe even more than that, are indeed completely asymptomatic, and they have the ability to spread. And there are like 6 different kinds of COVID or clusters of COVID. And as it moves further, I suspect there will be more than these 6. You can read on the slide as to the severity here. The first 3 are less severe, excuse me, and the last 3 are more severe. So if that gives you an overview of COVID, where do we go from here? Number 1, I think that there's going to be a normalization of COVID in 2022. We're starting to see that now. Most importantly is on the top bullet here, individual health versus COVID health, from mandates to protection of the vulnerable. And I hope vaccines will be available for all ages, hopefully by mid-year. There's going to be an expansion of the workplace role for COVID protection and for health, expansion of patient direct role with tests being done at home. And when we talk about public health, I don't know. I don't know whether it's going to be expansion or contraction, but we will see changes. To date, we saw this huge expansion of the role of public health, but a lot of our society work against that and say it should not, and it should contract from now. So overall, the future imperatives that I see is that COVID testing and future vaccinations, I believe, like flu, will move to the workplace. Telemedicine is fully integrated, partly because of the red box here, which is self-testing, becomes standard. And lastly, and I'm going to cross my fingers here and hope that we have lessons learned from this pandemic and that we use those lessons for public health and individual health for the future so we can do better as we move forward. So with that, let me say thank you and feel free to be in touch with me, and happy to follow up, and stay safe, everybody, stay well. Thank you.